Bismillah ar-Rahman ar-Rahim. In the name of Allah, the most merciful, the most kind. We start 5090 O-Level Biology, June 22, paper 6-2. And we're going to discuss how to do this paper in a befitting manner and to discuss the grade boundaries of it. As you can see here in paper 6-2, the grade boundary was sort of slightly lesser. It was 27 marks as opposed to 28 marks. And with B grade it was 23 marks. So it was a slightly difficult paper. C was 20 marks and in 6-1 it was 22 marks, 17 marks for D grade and 14 marks for an E grade. So now this is what I want you all to understand is that these grade boundaries are very essential for you, very important. When you make 3 careless mistakes, if you make, now you are going to get 27, but you made 4 careless mistakes, you go down to 23. Well, the careless mistakes are going to cost you a grade. And then you make 3 mistakes, you go into a C, 20 marks. Another 3 mistakes and you go into 17, D grade. So you just do, I've been checking papers these days of A-level students and I see so many careless mistakes that it really makes me very, very sad. And I feel that you all must be aware of this, that you cannot afford to make any careless mistakes. 509062, June 22. And this is for 1 hour. As I've told you before many times, you have to really manage your time very well in this. Enzymes are used in the fruit juice industry to help with the extraction of juice from fruit. Some students wanted to investigate the extraction of juice from some crushed apples. They were provided with 2 beakers labeled A and B containing the same mass of crushed apples, a solution of an enzyme, 2 measuring cylinders labeled A and B, 2 filter funnels and some filter paper. They followed these instructions. Add 2 cm cube enzyme solution to beaker A. Add 2 cm water to beaker B. So we have A and we have B. We have A and we have B. So the enzyme has 2 cm has been added to A. And water has been added to 2 cm water has been added to. So the volume is the same, but here we have added an enzyme and here we have added water. Use a clean stirring rod to mix the contents of each beaker. So they should be separate because in one we have got enzyme in one, so we must have 2 separate rods. Leave the beakers for 5 minutes. Place a piece of filter paper in each filter funnel. So you take a filter paper and you put it here in the funnel. And now this is the funnel here. And you've added the filter paper. Place one filter paper in measuring cylinder A and the other in measuring cylinder B. So we've got this here. We've got, you see a picture of it. After 5 minutes, fill the filter paper in A with the crushed apple, an enzyme solution from beaker A, and fill the filter paper in B with the crushed apple and water from the beaker. Start the stop clock and measure the volume of any apple juice collected in the measuring cylinders A and B every 2 minutes for 10 minutes. Every 2 minutes for 10 minutes. So start the stop clock, measure the volume of any apple juice collected in the measuring cylinders A and B every 2 minutes for 10 minutes. Now, whenever you're reading this question, imagine that you're doing this in the lab. And this will help you to then do this paper much better. Name a piece of apparatus suitable for measuring 2 cm cube other than measuring cylinder. Well, you could use a syringe. You could use a syringe or you could use a graduated pipette. I hope you've seen all these things. So a graduated pipette or a syringe, you could have used any of these. Some of the students' results are shown in the table. Time, total volume of juice collected. Now you can see this table. The diagram shows the juices collected in the measuring cylinders after 10 minutes in A and in B. Now it says complete the table heading and record the volumes of apple juice in the table. So complete the table headings. So total volume will be cm cube. And this will be either you wrote the whole of it minutes, or you wrote it in minute. MIN was also correct if you wrote that. So time in minutes, or MINS, or MIN was also correct, or MINS was also correct. And of course, so this got you 1 mark. Now you had to see what was this. So this was what? This was 22. And how much was this? This was 5, 6, 7, 8.5. So this was 22.0 and this was 8.5. So you completed this. Diagram shows the juices. Complete, record the volumes of apple juice in the table. Right? Use the data in the table to suggest why the use of the enzyme is an advantage in the fruit juice industry. Now, this of course is very important. Why is it important? Because the enzymes digest the cell wall and the other contents, which will then release larger volume of juice. So it will be faster or increased rate and more profit or more efficient or more money or more cost effective. So basically we want more juice because we are in the juice industry. So we want more juice out of the same fruit. So the same apples give you 20 ml, and then if you use the enzymes gives you 30 ml. Well, that's good for you. You have more juice, more money. So more profits, more money, more efficient, more cost effective. So for 3 marks, you can see here for 3 marks, you have 3 mark scheme points. So you had to give me all these 3 points to get your 3 out of 3. When you come out with the paper, you usually message me and say, oh, miss, my paper was very good. I think only 5 marks will be cut. No, 5 marks will not be cut. More will be cut because you will have made casual mistakes. So it releases larger volume of juice. It is more juice per unit time, more profit, more money, or more cost effective. I'm sure many students would just got 1 point out of this because they might not have worded it correctly. Even if they knew the answer, they probably wouldn't have worded it correctly. Now, coming to the D part of the question, suggest reasons for stirring the contents of beaker A and B. Why did we stir them with a glass rod? Mix or evenly distribute the contents. So you wanted to mix the contents. Why do you stir when you have a cup of tea and you put sugar in it? Why do you stir it? So that the sugar dissolves in it and it mixes the sugar and the tea. So to mix the contents, or you can say evenly distribute the contents. Now this is again a matter of more of your English rather than anything else. Why suggest reasons for using a clean stirring rod to stir the contents of beaker B? Because one has enzyme and the other has only water. So prevents contamination with the enzyme, prevents transfer of the enzyme. So if you in A is the enzyme and in B there is no enzyme and you're stirring it in A, and then the same rod you're stirring it in B, so the enzyme has been transferred from here to B. So prevents contamination with enzyme. So prevent the transfer of the enzyme from beaker A to beaker B. And then we come on to the E part of the question. Explain how the contents of beaker B act as a control. C for control, C for comparison. So we want to see that in A there was an enzyme present. In B there was no enzyme. So is there going to be any difference? With the enzyme, was it going to be more juice, or even without the enzyme, there was going to be a lot of juice? So that means, did the enzyme work? Should we use the enzyme? Because the enzyme costs a lot of money. So what happens without enzyme in B? Does the same overall volume or only one variable is changed? For comparison? So 3 points. Why do you use a control? For comparison, number one. And then to see if without the enzyme also is there the same volume of juice produced, then of course there's no need to use the enzyme. Now I want to explain this to you all because some of you are not understanding my point. I know it. Explain how the contents of beaker B act as a control. What happens without enzyme? Fine, this was 1 mark. To compare, second mark. But what does this mean? Same volume of crushed apples and liquid. You see, in A, you had the crushed apples and then you added some enzyme solution. So in B, now you had the same crushed apples, but then you added just plain water to it. So both will have the same volume. This is also an important thing to standardize it. If you did not add any water, then the volume would have changed. So you're just adding plain water to make the total volume the same. In one, you added an enzyme solution. So it must be some fluid. It must be some liquid. So same volume of the crushed apple and the liquid. And that is an important thing which you have to understand. Okay, now coming to part F. Use the information in the table to construct a line graph for the data for A and join your points with ruled straight lines. First of all, you've got to remember that we have to only construct a line graph for the data for A. So we go back to A and we circle A. Now the time in minutes, this always goes on this is the first column, always goes on the x-axis. So this will go on the x-axis and this will go on the y-axis. So let's select, here you've got 0 to 10. And here you've got the highest is 22. So you see on the graph that you've got to adjust this. It should go right to the end. So this is how I have decided to take the graph and the scale which I have chosen, because I want 10 to be coming right to the end and 25 to be coming right to the end here, so that I can use more and more of the graph. Now let's plot the points, and I've written these on this side. So 0 is 0 0.0, so you've got this here. Then 2 is 12.0. So this is 10 and 15. So two small squares are equal to 1. So this is 2 is 12. Then 4 is 18.5. So this is this. And then 6 is 21.0. So we've got it here. And then 8 is 21.5. And 10 is 22. So we got all these. I'm just trying to show you all this. I'm sure you all know how to do this. And then we have to join them with a line with a ruler point to point. When you join it with a ruler, I'm sure I haven't done it that well because I can't use a ruler on this. It's a little difficult for me to do it on my iPad, but I'm sure you'll do a good job of it. So join it with a ruler point to point. Because there was a 0 here, so then you have to connect the 0. But if there was no 0 and it started from 2, then you will only start it from 2. Then you won't do it. Then you'll only do these points. But there was a 0 in the table, so you would have to do that. You have to remember that. Now coming to a very simple part, but here is where careless mistakes will start. Use your graph to estimate the volume of juice collected at 5 minutes. Show you're working on your graph. Volume of juice collected at 5 minutes, 3 marks. Now what do you think are the 3 marks for? The 1 mark is for correct working on the graph. The second mark is for 19.5 or up to 20.5. It depended on your graph. And then the third mark is for cm cube. So 1 mark for you to show it on the graph, 1 mark for 19.5 or anything between 19.5 and 20.5, and the third mark is for writing cm cube. Now you can see here I've drawn this line, this green line 5, and then you see it here is nearly 20. So if you gave me this line here, this is at 5 minutes. At 5 minutes you got this, and it was 20 cm cube. So let's change it here. So according to my graph, it was 20 cm cube. So the fact that I drew the line, I got 1 mark for this. And then the fact that I wrote 20, I got 1 mark for this. And the fact that I wrote cm cube, I got 1 mark for this. Now it's very sad if somebody did not get these 3 marks. It's absolutely something in which you don't need to know any biology. It's just something that you have to get it from the graph. Now coming to the difficult part. Design an experiment based on the method used by the students to investigate the effect of the enzyme on the production of juice from different varieties of apple. Give full experimental details. And this was 6 marks. Now, this is something which is difficult. Yes, I agree with that. So, when it says different types of apple, now that's something which you know from the question. So, just say same mass of crushed apple used of different varieties, green apples, red apples, yellow apples. There are different species of apples. The green ones have got are very sour, and there are ones which are slightly yellowish, and then which are totally red. So different species. So same mass of the crushed apple used, then the same volume of the enzyme solution used. Then you keep the same temperature. What are you changing? You're changing the types of apples. Same temperature, then you mix the apple and the enzyme and you leave it for 5 minutes. Then you filter it, and then you measure the volume. Filter it, and then you measure the volume, and then you see, compare, and the juices produce. Is it more in one variety or less in the other variety? And that is how you'll get a comparison. So I've just worded it for you, different types, but same mass of each type, same volume and concentration of enzyme used, same temperature. Mix the apple and the enzyme and leave for 5 minutes. Then using a funnel and filter paper, filter it. Filtering time should be the same. If you leave it for 10 minutes, all of them should be left for 10 minutes. And then you compare the volume of juice produced. Was it more or less with different types? Now, remember, this is when you are sort of doing this experiment, you should consider the one they've given you. They've given you some rough idea on that. They've just changed one thing. In that, there was just one apple. Here, now you'll have different varieties of apples. Now, same temperature you can write nearly in every experiment. And then the fact that you have to mix the apple and the enzyme, that you should have learned from the part of the earlier part of the question, that you have to mix in the apple. So because it said give full experimental details, so think that as if you're doing this in the lab. How would you have done this in the lab? So that is one of the very important methods by which you learn how to do this 6 mark design and experiment story. Then coming to the last part of the question, a student tested some apple juice for the presence of reducing sugar and observed a red color. Name the testing solution the student used, state a temperature at which the test was carried out, and a conclusion for the student's observation. Now, testing solution, as you all know, is Benedict solution. I'm sure everybody should know this. There's nothing that. So Benedict solution, as solution they have said, so if you did write it or didn't write it, didn't really matter. And then what was the temperatures? Anything between 80 degrees Celsius to 100 degrees Celsius. So if you said 80 degrees, that was fine. But what was allowed was anything between 80 and 100. You should just write one, don't write a range. Then the conclusion was that it contains reducing sugar. Now, what will a large number of students write? Just contains sugar. Well, it's wrong. It's not sugar. Sugar are two types, reducing sugar and non-reducing sugar. So you just can't say contains sugar. So you had to be very clear and say reducing sugar. So this was the 3 marks. The 1 mark for this, one for this, and one for this. So this was a total 27 marks. So it's a 40 marks, total 27. Question 2. The photomicrograph shows some human blood cells. We have some labeling C, D, E, and F. In the space below, make a large drawing of the cell labeled C as it appears in the photomicrograph. So basically, I always say you just imagine as if you are tracing it out. So there is a line outside it, and then you have to make this type of a diagram inside it. Well, that's it. So you just have to draw, but it has to be a larger drawing of it. So you just have to draw it. I'll try to do a decently job of it, but it doesn't really need a lot of drawing skills. I know that. Now you have to understand is that there was this E to F. There's E to F. So it says this should have at least been 6 centimeters. So this area should have been at least 6 centimeters. It just means that they want you to draw a large drawing of it. This is 4 marks. This is the space. In the space, you must occupy 3/4 of the space. So clear, continuous, and clean outline of the cell and nucleus, and no shading anywhere, and correct orientation. Well, got you your 1 mark. E to F, at least. This is E to F. I'm not saying that you write this on the paper. This should have been at least 6 centimeter. So this is what we need to understand, that this should have been at least 6 centimeter. So you don't write the E to F, I was just explaining that to you. Then there should have been a U-shaped nucleus occupying most of the cell and touching the edge of the cell on the right-hand side. I'm just going to show this. This is the one which is touching the cell on the right-hand side. So U-shaped nucleus occupying most of the cell and touching the edge of the cell on the right-hand side. So this is the right-hand side. Then upper part of the right-hand side, all nucleus smoothly rounded. So this was also, it should have been smoothly rounded. This part should have been round, because you see, if you look at the diagram, this is also very round here. So upper part of the right-hand side should have been of the nucleus smoothly rounded. So this part, upper, this one, this part, and this part should have been round. So this would have got you your 4 out of 4. Now it says identify the cells labeled C and D. Now C is a white blood cell, and this is a red blood cell. Now this was only for 1 mark, so it wasn't for many marks, but this is very clear that you all had to underline it. So C and D. So this is, D was a red blood cell and C was a white blood cell. Or you could have said a phagocyte. They haven't said that, but you could have said anything. White blood cell is fine because it said the type of cell. Then it says C part, complete the table by describing 3 visible differences between C and D. So you come back to C and D. Let's go back to C and D. Let's see here. C and D was what? C is larger, D is smaller. C is larger and D is smaller. Yes, fine. We could do that. Then C has a nucleus, D has no nucleus. Then C is granular and D is not granular. Then C is not round, not circular. D is round and circular, or round or circular. So just look at it and then think of the points which are comparison: larger, smaller, nucleus, no nucleus, granular, not granular, round, not round. So larger, smaller, has nucleus. So these are 4 mark scheme points, and you were expected any 3. So it's not that you write all the 4, so you'll get the 4 marks. So it's only a 3 that you had to write. Now it says draw a straight line on the photomicrograph to join E and F. Measure and record the length of this line, length of the line E to F, and in millimeters. Now length of the line was anything between 15 and 17. So 15 millimeters. Then you had to, then on your drawing, draw a straight line at the same location as the EF and measure and record the length of this line, length of the line on the drawing. So you had to do this on your drawing now. Now it says you had to draw a line on the photomicrograph and then you had to draw a line on your drawing. So if you look at here, I've done this here, E and F, I've drawn a line here. You can see that here, the orange line. And then I've drawn it on the drawing. And this line, which I've drawn, is 8.5 centimeter or 85 millimeter. So this line, I've measured this line E to F. This is the line that I've measured from here to here. Now, similarly, the line here to here, E to F, was 15 millimeter. And here it is 85 millimeter. So I've written that and I've shown the lines on it because the 2 lines for this part 2, there were 2 marks. For part 2, for the 2 marks, it is line drawn in the correct position and measurement. But here there's another 2 marks here now for this. Use your measurements in 1 and 2. So you're only going to talk of 1 and 2 and calculate the magnification of your drawing compared to the photomicrograph. So the photomicrograph is 15, yours is 85. So 85 divided by 15 will give you the magnification. And you give your answer to 1 decimal place. So how do we measure this? So this was 85 and then divided by 15. So you see the 2 marks are: number 1, you showed me this working, 85 divided by 15, this got you 1 mark. And then the answer is 5.66, but it said only 1 decimal place. So I'm not going to write this. I'm going to write 5.7. Magnification, there's no units. X means how many times is it magnified. So the 2 marks I would get, one for this working, showing the 85 divided by 15, and the other one was believe for 5.7 because it says give your answer to one decimal place. Actually, it is 5.66 was the answer coming on the calculator, but then I have rounded it off to 5.7. That finishes this another paper 6, and I hope this is helpful, and please go through it now. Don't wait for it till the end of the month or later on, because you must finish these papers and do more of them so as to understand them better. Thank you and best of luck. Please do write your comments and please do let me know how these are helping you and which other papers would you like me to do. Thank you very much.